Hey guys! Today we're going to talk about how drugs are developed and the clinical trials that make sure that they're safe.
Firstly, we're going to talk about drug development. Initially, drug manufacturing companies start with various compounds to develop a specific drug. Then they must choose the best or most effective drug, narrowing down the range of compounds.
To test for safety after being produced, the two types of preclinical research are in vivo and in vitro. In vitro means in glass studies, and they're performed with microorganisms, cells, or biological molecules. In vivo means in life, and these studies are those conducted in whole animals.
Next is clinical trials. The MHRA must authorize clinical trials. These are sponsored by the NHS, the Medical Research Council, and the Department of Health, as well as charities and pharmaceutical companies. Phase 1 is carried out on patients who volunteer to be tested on. Phase 2 is where the drugs are tested on a small number of patients with the disease. And then finally, Phase 3, a number larger of patients are tested on.
If the drug isn't found to be toxic, these trials provide the information on how much of the drug is safe to give, what the side effects are, and how effective the drug is. And this process can take up to 10 to 15 years, but this time span varies.
After clinical trials, the MHRA can now approve the drug for pharmaceutical marketing. It can only be licensed if high standards of safety are met throughout the whole development and manufacturing of the drugs. Drug companies take out patents for each new drug they discover, so only that company can produce the medicine and make a profit, which lasts around 20 years in the UK. Once the patent expires, other companies can now produce their own version of the medicine for cheaper, because they don't have to go through the whole research process. People using the drug after clinical trials, so once it's on the market, can report it to the yellow card, and if they personally find that there is harmful side effects.
Research is important in medicine. For example, research on the effects of genome editing could help us to find solutions for many common diseases like cancer and diabetes, or show us if this method is too dangerous. It's also a part of evidence-based medicine, which involves using the most recent and reliable evidence from research to give good healthcare for patients. It can show us the effect of lifestyle drugs and disease on our population, and it can help doctors and scientists provide advice on drugs to improve people's quality of life. There are many ways of carrying out research, some being more effective and reliable than others.
This is the pyramid of research. The top of the pyramid shows the most useful methods of research, and the bottom shows the least useful. So at the top there's meta-analysis, and this involves combining data from multiple studies. It's more representative of the population, and it helps eliminate bias and anomalies. Second is blinded and double-blinded RCTs, and they test drugs eliminated bias, where the participant doesn't know which drug or placebo is being tested on them, and nor does the doctor that is prescribing them.
Prospective cohort studies involve studying links between risk factors and health outcomes for the future, assessing future health risks such as future increase in skin cancer. Retrospective cohort studies look at the past cases of disease and risk, and they tend to be more biased the further back in time they go. Case series or case studies are studies of people with a known treatment or disease and assessing the outcome. And expert opinions and anecdotal articles could be biased or come from unreliable sources.
Thanks for watching our video.